Hello, welcome back to another DNA session for DNA learning session. Magandang gabi, bayan. I'm kidding. So now let's start with the relationship between DNA structure and replication. Hopefully my mic works. Let me just, yep, double checking finished. It works. So let's start with a famous excerpt from the 1953 paper by Watson and Crick. It outlines the discovery of the double helix structure of DNA. The quote is, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So yeah, and the quoted sentence refers to the implication of complementary base pairing between nucleotides in DNA. And yeah, it's the discovery was critical in understanding how genetic material is stored, replicated, and inherited. All right. And DNA replication occurs through a process where the parent strands, each strand of the parent DNA double helix serves as a template. We mentioned that before, and the parent DNA consists of two strands, the S and S strand. It's in opposite directions, so that's why you have different new synthesized strands, and they're both synthesized in 5 prime to 3 prime direction. It's just that when you open the bubble, it's coming from a different area, so that's why you have the leading and lagging strand. And again, it occurs in the 5 prime to 3 prime direction. It's very essential and important. It's a fundamental aspect. And you have an enzyme called DNA polymerase, which adds to the incoming nucleotides. It always attaches them to the 3 prime strand. And it is according to ATGC. So you have the purines, right? Adenine and guanine. And then you have your pyrimidines, cytosine, and thymine. And the purines and pyrimidines would partner up with each other, with adenine pairing up with thymine, and guanine partnering with cytosine. And the process would start with the primer. Okay, the primer strand, and that would provide a starting point for the DNA polymerase. And the incoming nucleotides are in the form of, well, DNTPs, deoxynucleoside triphosphates. And that would contain 3 phosphate groups added one by one, and as each nucleotide is added to the strand, the 2 phosphate groups are chucked off, and that would release the pyrophosphate and providing the energy for the reaction. And that will ensure that the DNA strand goes through the process as 5 prime to 3 prime. Let me just draw the structure of the ATP really quickly. So you have the sugar OH over here, CH2, and then you have OP, 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 and then another O, oxygen, double bond oxygen. Whoops. Yep, that's correct. Double bond O minus, another O minus, minus over there. The first phosphate would attack the OH at the third position. 1, 2, 3, 4, 5 of the one before. Whoops. OH. It attacks that. Or, I mean, the other way. The oxygen attacks the alpha one, alpha, beta, gamma. And that would lead to the pyrophosphate being formed. That's the PI, pyrophosphate. Okay, important. Next is DNA replication is semi-conservative. It means that it would need, well, meaning each of the two daughter strands consist, oh, wait, one more time. It's the two daughter molecules contain one from the old parent and a strand that's newly synthesized. That's how it is semi-conservative. And in the replication process, the double helix unwinds and the two parental strands are separated. And again, each parent strand would serve as a template. And this model of replication was confirmed by the Mendelssohn style experiment, and it's a fundamental principle of DNA replication. Yes. The DNA replication fork is the structure formed during the process of DNA replication, and it's a region where the DNA is unwound, allowing the parent strands to be separated so that they can serve as the templates. And DNA polymerase, it's a key enzyme that synthesizes DNA. It adds nucleotides to growing DNA strand using the single-stranded template as a guide, and it recognizes the base pairs, and then it between template strand and the incoming ones, and it would attach it. Once the correct nucleotide is positioned, DNA polymerase catalyzes the formation of the phosphodiester bond. And then you now have the correct form. Take note that the pair is from the 3' end of the primer and the 5' end of the next one. Yes, that's how it happens. It needs a 3' hydroxyl end as the substrate, and the pyrophosphate is let go. Note, there's another thing important. You have helicase, which unwinds the DNA ahead of DNA polymerase. So helicase is the first one, I think. And unwinding is important because how can you replicate it if you don't even have the starting area for it? Okay. You unwind it, you allow each strand to serve as a template, and the way you unwind, helicase uses energy from ATP each time it moves. An inorganic phosphate is let go, and that breaks the hydrogen bonds between them, etc. And you have the single-stranded binding protein that exposes the template strand. That is crucial because it maintains the stability of the single strand. And how? Well, as the DNA helicase earlier unwinds it, the single-stranded regions would want to go back. So think of the SSBs as the clips to prevent them from going back. It helps stabilize the single-stranded template. And note that you have a sliding ring, or which would keep the DNA polymerase from falling off. So what is that? I know. But the sliding clamp or ring, it's another important component, and it ensures that the DNA polymerase stays attached to the template strand while synthesizing the new DNA strand. And the clamp is a ring-shaped complex. First, there's two things. You have the clamp loader and the sliding clamp, okay? First, you have the clamp loader binds to ATP and it opens up the ring. Again, clamp loader binds to ATP and it opens up the ring. Actually, wait, let me just adjust the location, the size of the 2048. Here you go. Just so the score is clear. Okay, good. Yes, again, clamp loader attaches to the attaches to ATP. The sliding ring is forced open, and the sliding clamp then encircles the DNA. And then once the clamp is placed, ATP is hydrolyzed, and that would lock the ring. And then after that, the clamp loader is now gone. And now you have the sliding ring there. Next, DNA polymerase would bind to the sliding clamp, the one that clamped inside. And then that would ensure that DNA polymerase can continue working without falling off. Yeah, and then the clamp loader that was let go, it would just come back when it's needed already. Okay, so again, you have the sliding clamp and the the clamp loader and sliding clamp. Okay, the clamp is essentially the ring. So first you have the clamp loader, which binds to ATP and also brings along the sliding clamp, which is opened up. Sliding clamp hugs the DNA. ATP is hydrolyzed and clamp locks itself around the DNA. After the ATP hydrolysis, clamp loader is let go, and then DNA polymerase comes along, and then now it can do its work without falling off. Topoisomerase meanwhile uncoils and detangles the DNA around, because if you unwind it using the DNA helicase, then the back part would be clogged. But now you have the topoisomerase, which we talked about earlier. The topoisomerase would be first. It has types 1 and 2. Type 1 would cut one strand, but type 2 cuts 2 strands. And the next one just talks about that one. Topoisomerase binds. We're talking about type 1. Topoisomerase binds, and then it would use the tyrosine residue to break the phosphodiester bond. How topoisomerase works. Well, it would use the tyrosine residue, OHCH2, and then OH at the bottom, CH2, benzene ring, and OH. After the tyrosine cuts the phosphodiester bond, the cut allows the DNA to rotate around it, and you reform the break. Now, that is how the detangling works. And then you have topoisomerase 2, which would cut both, where it recognizes the entanglement, and when two circular you have the two circular DNA, you have the gate. Okay, so then let's start. This is a little bit different. Recognizes the entanglement, and then topoisomerase isomerase 2 binds to it. Okay, you have two rings like that, and then it would bind in the middle, open up, and it covalently attaches to the opposite strands. It creates a temporary break between one strand, allowing the other one to pass through, and then it restores it. Okay. Next is the problem between the lagging and leading strand. So the leading strand is synthesized continuously in the 5' to 3' direction. And it follows the direction of the replication fork. Meanwhile, the lagging strand, it is synthesized in the opposite direction, aka even though it's 5' to 3', it has to do it piecewise. and it must work in short bursts. So that is why. Again, if I try to draw it, you have this thing open. 3 prime, 5 prime, 5 prime, 3 prime. If the fork is open here, this one's the continuing strand. Meanwhile, the 5 prime, 3 prime goes like this. So this is the leading at the top, and the lagging is at the bottom. Alright, RNA serves as primer on the lagging strand. And the RNA primer, well, they're both used as the RNA primer. RNA primer would serve as like the starting point. And since DNA polymerase can only add nucleotides to an existing 3' end, that's why the RNA primer is important. DNA polymerase. So again, DNA replication, you have RNA primer, DNA polymerase, which would add DNTPs and etc. If you want to compare it with PCR primers, in the sense that they both provide a starting point, but a bit there's the similarities is they provide a starting point, and PCR primers are also short sequences that provides 3' hydroxyl group to the DNA. But the main difference is the types of primer, okay? Primase makes it in the lagging strand, but in PCR, polymerase chain reaction, the primers are synthetic DNA sequences, and they are not RNA synthetic. So they are made to be complementary to the specific sequences that you want to target. And it's chemically synthesized. Note that in PCR, you have the forward and reverse primer. And these primers define the region of DNA to be amplified. I mentioned that before. In natural and in removal and replacement, normally the RNA primers are removed and replaced by DNA pol 1 in prokaryotes. And in PCR, the primers stay. And after the amplification cycle, the resulting DNA contains the exact sequence between the two primers. And just a reminder for the lagging strand, that is the Okazaki fragments. The DNA polymerase adds nucleotides to the RNA primer, but the rest of the stuff is the same, and except for the DNA ligase where it tapes it up, it seals the nick. And how does it seal the nick? Well, so at the last part, at the nick, there is a 5' in the one strand and a 3' hydroxyl group on the other. So that's the nick. Their ATP is used by DNA ligase, and the enzyme would activate the 5' phosphate of the nick, and by forming a covalent bond to it, it creates an AMP linked intermediate. After the AMP is attached, DNA ligase catalyzes the formation between the 5' phosphate and 3' OH, which seals the break. Essentially, it would just connect the 5' and then the 3' OH. Okay. And then AMP is then released. But just to try and sum that part up, you have the DNA helicase, which unwinds single strand binding protein, would keep the zipper from rezipping itself. Primase would make the RNA primer, the DNA primer. Wait, one more time. Primase would make the short RNA primers on the lagging strand and provides the 3' hydroxyl group for the DNA polymerase to come in. And you have the leading strand, DNA polymerase is there, synthesizes in the 5' to 3' direction, same direction as the replication fork. Lagging strand is the opposite direction, and that's why you have short fragments such as Okazaki fragments. You have the sliding clamp and clamp loader. The clamp loader would come in with ATP, and the sliding clamp would open up and hug the DNA. And then after it hugs it or goes around it, ATP is what's the term? ATP is used up. After ATP is used, that clamp, the sliding clamp would then lock in, and then clamp loaders let go. DNA polymerase can come in. In the Okazaki fragment, since it is fragmented, you need the DNA ligase to seal it using ATP. And note, DNA replication has a very low but finite error rate. And in the first step is the 5' 3' polymerization, right? And the error rate is 1 in 10 raised to 5. And so you have the proofreading from 3' to 5'. It's called nucleolytic proofreading. And that would reduce the error rate 1 into 1 in 10 raised to 2. 1, okay? 1 in 100. And then you also have stand directed mismatch repair. Just anything that were missed by proofreading would be like the incorrectly put nucleotides would be just fixed by the mismatch repair system. So the combined error rate overall is just 1 in 10 raised to 10. So that would help it really reduce the error. Again, the DNA polymerase can do it. It has 2 jobs. It's moonlighting. It can do polymerization or adding nucleotides to the growing strand, but it also edits it after. So first there's the P part and the E part. It would edit it there. If an incorrect nucleotide is added, it goes backwards just to remove it and replace it with the correct one. It's an important part because it will help provide the high fidelity. And for its steps, you have a couple of steps. One second. So it extends the DNA by using DNA polymerase, right? So let's say let's say in the beginning you have an adenine, but it accidentally puts cytosine. So that's wrong. So for its exonuclease activity, the 3' to 5' exonuclease activity is triggered, and the enzyme chews back from the 3' end. It just eats that up. And then now you have a correct base pair. But if you because it's now empty, and you can add the correct one. And that's an important proofreading. Stand directed mismatch repair is you bind the mismatch proteins, mismatch proofreading protein, and it slowly scans. That's the mute S and mute L. Okay? Those are the two proteins. And the Miss Mute S and Mute L would scan it. And the wrong strand is excised by the Mute S and Mute L. Just removes that. And then DNA polymerase fills the gap. So the difference between the 3' to 5' exonuclease activity versus the mismatch repair is a bit obvious. So you have 3' to 5' will just chew up from polymerase and from the polymerizing end of the DNA polymerase, and you have the E excising part. It would just get that specific base pair would be eaten up. Meanwhile, the strand directed mismatch repair, you have the proteins mute S and mute L, which would drop the specific strand, and boom. And reminder, the initiation would start at origin sequences. Okay. And that would be replication origin, and that's where helicase comes in, and blah blah blah. Small bacterial genomes have a single origin of replication, and that's where the bubble starts. Meanwhile, the large eukaryotes would have more bubbles. And for example, we have a picture here where you can label it with thymidine for 10 minutes. It is radioactive. And the way thymidine works is it incorporates into the newly synthesized DNA strands. So you can track the replication. And that is a silver grain in the bubbles. That's a technique that I need. Oh, labeling with 3' and no 3H thymidine. 3H. Where is my papers? Technique is 3H thymidine. Labels newly synthesized DNA strands. Shows up as silver. Okay. Next is you have a dedicated initiation proteins. You have dedicated initiation proteins that assemble at the DNA complex at the origin. And note that the origin is an AT-rich region, which is easier to separate because it only has 2 bonds. And then after it breaks up, you have the helicase and etc. Also, there is the part where methylation sensitivity prevents the premature. Where's my sticky note? Okay, found it on my sticky note. Note. Where was it? Yeah, methylation sensitivity prevents premature reinitiation of DNA replication by controlling the timing of it. So, initially, it is fully methylated. Okay? And then that would allow DNA replication to begin. But after replication, it is just hemi-methylated. It's hemi. So one strand is methylated, the other is not. So that's what, and the hemi origins are resistant to reinitiation. So to initiate, you need to be fully methylated. Note again, to initiate, you need to be fully methylated. Okay? Over time, the origin becomes methylated again, so that's why it can start up initiation again. phosphorylation of the origin replication complex would prevent premature reinitiation in eukaryotes. So if we want to check uh, this is talking about the cell cycle, okay? This process explains how phosphorylation of the origin replication complex in your eukaryotic cells would ensure that DNA replication only occurs once per cell cycle. And in the G1 phase, the origin replication complex would bind to the DNA along with, and the ORC would recruit a couple of things, okay? ORC would take in CDC6, CDT1, and MCM helicase to form it. And then, once the cell goes into the S phase, ORC and MCM are phosphorylated, which would then activate helicase, and that would lead to the displacement of CDC60 and CDT1. And then and the phosphorylation also recruits DNA polymerase, I mean. So that's how it all starts. So again, let's mention it, okay? I mean, let's go through it again. During the G1 phase, the origin replication complex would, this is a really good one because in the slides for the G1 phase, in the cell cycle one, I don't think I saw this concept. I'm gonna have to sticky note it. Yeah. So during the G1 phase, there is the origin replication complex which comes in, recruits CDC6, CDT1, and MCM helicase. Okay. After that one, the ORC and MCM are phosphorylated. After it is phosphorylated, that would activate the helicase. Also, the CDC6 and CDT1 are let go. The phosphorylation of those would also lead to the recruitment of DNA polymerase. So that's how it all starts. And you have a telomerase, it is a dedicated riboprotein which would maintain telomeres, aka the ends. So the telomerase RNA would serve as a template for the synthesis of telomere DNA. Again, the telomerase protein would make it. It has a couple of things. Telomerase protein has fingers, which would help position the RNA template. The palm would contain the active site for the telomerase enzyme, which would add the nucleotides. And you have the thumb, which stabilizes it all. Okay, the fingers would position it. The palm would be where it actually happens. Thumb would stabilize. That's the telomerase protein. And it makes the template, the RNA template, and also extends it. Telomerase again uses RNA template to extend the telomeres at ends of chromosomes, and it first binds the 3' end of the incomplete newly synthesized strand using the RNA template, and the telomerase would extend it by adding the different repetitive sequences. And once it is extended, DNA polymerase completes the strand by synthesizing the lagging strand, more of it. And then The naked ends of telomeres are really protected in a loop, in a T loop, which prevents the naked chromosome ends from being degraded. And there is a 3' overhang at the end of the telomere. This overhang loops back to form a T loop. So the 3' overhang loops back to form a T loop, and that is now, it hides the chromosome end. And there's also the detail where the telomere length is carefully regulated throughout a cell's life cycle. And the longer telomeres would be shown are shown to be more stable, and that's why you really try to protect the ends. And as the cells divide, the telomeres would shorten, and with each division, because each division you lose there's the inability of DNA polymerase to fully replicate the chromosome ends, and that would lead to the gradual loss of it. All right. Next is, ooh, we have the DNA transcription next. Yeah, and we can save that for in a bit. Should I? Yeah. All right. That's all for the DNA replication. Thank you for watching. Bye.